the effect of UVC radiation on the germination rate, time till germination, and growth rate of Raphanus sativus by James Montague. Introduction. There are three different bands of UV radiation, A, B, and C, which are divided by wavelength, which is seen in Table 1. UVA is the longest wavelength from 400 to 315 nanometers. UVB is the mid-range from 315 to 280 nanometers. And UVC is the shortest from 280 to 100 nanometers. Shorter wavelengths carry higher amounts of energy, but aren't able to penetrate through solid matter as effectively. This is seen as Table 1, or Figure 1 rather, where on the x-axis is the incident photon, or the wavelength, and on the y-axis is the amount of energy that wavelength carries. And you can see as I've added the three bands of UV radiation, UVC in the red carries substantially more energy in the shorter wavelengths. Peak absorbance is a wavelength at which a molecule or object absorbs energy from photons most efficiently. DNA has a peak absorbance of 260 nanometers, and proteins and lipids are around 280 nanometers, but it varies from the exact protein or lipid as they're different. Cellular death and dysfunction, as well as genetic damage, are all common results of UV exposure. My research question was, how does exposure to UVC radiation affect the growth rate, germination rate, and time till germination in radish or sativus? My hypothesis was, if there's an increase in levels of UVC radiation exposure, then there will be a decrease in the germination and growth rates and an increase in the average days until germination. For my methods, research was conducted at St. Paul Academy and Summit School in St. Paul, Minnesota by me. In two groups of radish seeds, R. sativus received exposure from a 25-watt, 254 nanometer UVC bulb. One was for 5 minutes, which resulted in 7.5 kilojoules per meter squared, and the other for 10 minutes, which resulted in 15 kilojoules per meter squared. The 254 nanometers is very close to 260, and also somewhat close to 280 nanometers for the peak absorbance of DNA and lipids and proteins, respectively. 150 total seeds were planted, 50 from each from each group. So 150 for high radiation, 10 minutes, 50 for low radiation, 5 minutes, and 50 were planted as control, which received 0 minutes of UVC exposure. Seeds were grown in germination trays with cotton strings acting as the irrigation system. This is seen with the irrigation system of the strings provided for constant watering in the trays. And then Figure 3 shows the completed lab setup with all the seeds, all 150 planted in the dirt. Plant heights plus or minus 0.5 millimeters were collected with a millimeter ruler, and germination data was collected every 3 days over a 35-day period. Air temperature, watering, and light were all kept constant throughout the study, and one way ANOVA, as well as two-tailed t-tests were used for statistical analysis to find significance in the data. Figure 4 shows the radish seeds after growth and after germination at the end of the project after 35 days of growth. They were grown in the Sun Garden Sunlight Tier 3-tier growth center to provide 12 hours of light constantly. Results. A positive relationship was found between high levels of UVC exposure and germination rate. Germination rate. The 10-minute group had a significantly higher germination rate than both the 5-minute and 0-minute groups. Two-tailed t-test where P was smaller than 0.05. There was no significant change found between the 5-minute and 0-minute groups. Two-tailed t-test where P was greater than 0.05. This is seen in Figure 5, where the 10-minute group has significantly higher amounts of seeds germinate than both the 5-minute and 0-minute groups, but there was no significant change between the 0 and 5-minute groups. Mean time till germination. There was no significant difference found between the 10-minute, 5-minute, and 0-minute groups, ANOVA, where P was greater than 0.05. And then as seen in Figure 6, the 10-minute group, as seen in blue, had a more normal distribution, whereas the other groups, both 5-minute and 0-minute, as seen in orange or yellow and red, had a higher percentage germinate on one side, or specifically the first side, so the more normal distribution was only seen in the 10-minute group. Growth rate. There was no significant difference between the mean growth rates of different variable groups. ANOVA, where P was greater than 0.05, this is seen as both the means as well as the standard deviations and standard errors were all pretty much the same. Discussion end for future studies. The hypothesis of the study was not supported, as high levels of radiation has a positive impact on the germination rate and no significant impact on the growth rate or time until germination. Higher energy UVC rays may have caused damage to the seed coating, lipids and proteins, or caused genetic damage. And this is because the peak absorbance of 260 or 280 nanometers for the proteins, lipids, and DNA is very close to the 254 nanometers image from the bulb. The weakened seed coating would allow for water to fill the seeds faster and for the embryo to break the seed coating easier. This meant that more seeds from that group would have germinated. But the organic mass of the seed coating would have blocked the rays from penetrating further and prevented actual genetic damage or damage to proteins and lipids within the seed or within the embryo, which would have made it so it did not affect the growth rate, which was found. Fewer seeds would be needed as a result of this from the higher germination rates. This means it would be more cost-effective planting for farmers, as they would have to actually purchase fewer seeds for the same amount of crops. And there would also be less organic waste and better water conservation, as fertilizer and water would not be wasted on seeds that would never actually sprout or become crops. It would also be easier transportation of seeds, both terrestrially and interplanetary. Fewer seeds would take up fewer space, and for interplanetary, less shielding would be needed for the storage for seeds, as seen in the study, certain amounts of UVC radiation or UV radiation in general can actually help the germination rate and would not affect both the growth rate and time to germination. More research is needed, however, on the adverse health effects of high radiation dosages. This is because there were some plants that either were abnormal colors or had different numbers of leaves for the radiation groups. Additional research is also needed on the genetic damage and implications caused by the radiation of seeds. This was not studied in this study. However, more research is needed on this, as genetic damage is important in terms of longevity of that plant species, and also in whether or not damage is actually done to the plant, and also whether it be a reproductive capability. So that's important in terms of long-term agriculture. For acknowledgments, I would like to give special thanks to Dr. Gary Gardner of the University of Minnesota and Dr. Julia Massa of NASA for their guidance over the development of this project. And this is my bibliography and references. Thank you.